my name is Birce. I am a graduate student in Dr. Thomas Hunn's lab at Ohio State University in the Department of Biomedical Engineering. So my poster today is exploring how increased CAM kinase activation of late sodium current alters calcium dynamics in atrial cells, and it's a mathematical modeling study. So the heart is an electrical mechanical organ, and that is to say that when the heart is electrically activated, that's what synchronizes the mechanical contraction of the heart. And so we're interested in how this electrical activity that goes across the heart synchronizes different cells and can remain consistent. And when it's not consistent, that can lead to problems such as heart failure and other terms of cardiac disease. Now, that electrical activity on the whole heart level can also be seen at the single cell level, and it's due to the movement of ions back and forth across the cell membrane that cause a change in voltage potential for the cell. With that change in voltage potential, that causes the cell to eventually contract. And so there have been these mathematical models that are sets of ordinary differential equations that describe the movement of these ions back and forth across the cell membrane. And the reason we're interested in mathematical models of electrical cell activity is because it allows us to integrate small amounts of data on different ion channel populations and then study complex biological systems. So our lab is specifically interested in one type of cardiac disease called atrial fibrillation, which affects more than 3 million people per year in the United States and is characterized by rapidly contracting random chaotic atria of different patients. So atrial fibrillation is highly heterogeneous. Patients with atrial fibrillation have a high morbidity and mortality. They're at 5-fold increased risk of stroke. And we're really interested in understanding what the complex mechanisms are that lead to atrial fibrillation for our patients so that we can understand how to better treat it. Now, we've seen that late sodium current, which is a small persistent current that persists throughout a cardiac cell's electrical activity cycle, has been shown to be really important in the context of atrial fibrillation. And late sodium current blockers, such as ranolazine, have been shown to suppress atrial arrhythmias. We've also seen that late sodium current in ventricular myocytes is increased in human heart failure. And we'd like to understand whether suppressing late sodium current alone is enough to suppress atrial fibrillation, or if secondary effects on other ion channels are important in atrial fibrillation as well. So our lab is interested in exploring late sodium current, and we're interested in understanding how a protein kinase called CAM kinase affects late sodium current in the cell. So my first step was to take CAM kinase and late sodium current and incorporate it into a model of a human atrial cell. So here you can see a model of an existing cell and the formulations that I added. So CAM kinase is targeting my late sodium current and a diverse array of downstream targets such as the L-type calcium channel and phospholamban and the renin receptor. And what we wanted to do is, once I incorporated these formulations into the mathematical model, then I wanted to test to make sure that these formulations were correct in the model, fit it to my experimental data and validate it, and then play with my model to understand how late sodium current dynamics are affecting the stability of the atrial cell. So here is some of the data that I fit. This is experimental data that is on the late sodium current as well as the behavior of CAM kinase activation of the renin receptor. Next, what I wanted to do once I incorporated these equations was test to make sure that my formulations had not deviated my atrial cell model too much. And so here you can see over a range of pacing frequencies, the duration of a single cell's electrical activity in my simulation did not deviate too much from what we see experimentally. Next, I wanted to look at CAM kinase phosphorylation of two of its main targets, late sodium current and the renin receptor. And so looking forward, we're going to focus on 100% phosphorylation of late sodium current and the renin receptor. So here are steady state transients for voltage and calcium, CAM kinase and sarcoplasmic reticulum calcium in each of my cell models. And what we've seen is that when CAM kinase phosphorylates 100% of my late sodium current, I get this increased voltage duration for my action potential. I get increased calcium dynamics, and I get increased CAM kinase activity. And in contrast, when it phosphorylates 100% of my renin receptors, I do not get that activity, and I get a little bit of decreased activity. We sort of expected this result, but this actually ended up being much more emphasized than what we expected, and so it was a bit of a surprise for us. Next, I wanted to look to see how CAM kinase phosphorylation of late sodium current and phosphorylation of the renin receptor affected ion homeostasis and steady-state CAM kinase activity. And when CAM kinase phosphorylates the late sodium current, I get this increase in my calcium dynamics, an increase in my sodium concentration accumulation, and increased CAM kinase activity, suggesting that when CAM kinase phosphorylates late sodium current, it goes back and into a positive feedback loop and then increases CAM kinase activity again. I did not get that kind of behavior when CAM kinase phosphorylated 100% of my renin receptors. In fact, I got decreased behavior in that cell model. And furthermore, when I looked at the behavior of my renin receptor, I saw that CAM kinase phosphorylation of the renin receptor and late sodium current affected my renin receptor function and my calcium flux. So while CAM kinase phosphorylated 100% of renin receptors in one of my models, I actually increased my open probability of the renin receptor, so increased the amount of calcium that was going through this protein by increasing my late sodium current. Next, what I did is look to see whether these cells were susceptible to any kind of abnormal dynamics that would make them susceptible to atrial fibrillation. Those kinds of abnormal dynamics are alternans, so alternating axial potential durations, or active depolarization, so you get a peak in your voltage when you're not supposed to get a peak. And I simulated a whole set of different mathematical models where I'm either phosphorylating 100% of my late sodium current or 100% of my renin receptor, or I've crossed them. And I've seen that I get the most amount of active depolarizations and alternans when CAM kinase has hyperphosphorylated my renin receptor and my late sodium current. And the model that was most protective against atrial fibrillation was when CAM kinase phosphorylated 0 of my late sodium current and my renin receptor channels. Next, I did a parameter sensitivity analysis, and I think this goes back to understanding why I'm doing mathematical modeling in the first place. This technique allows me to perturb all these different ion channels and run 500 different simulations and then see how all of these different cell models are behaving differently and be able to link the behavior of one ion channel and what its impact is on a measurement such as the amount of sodium or the amount of calcium or the whole action potential duration. And what I've seen here is that when CAM kinase phosphorylates late sodium current, that contributes to an intracellular ion homeostasis, and that's why we get these positive bars here on the left. In cases where I have increased beta-adrenergic stimulation, so I have applied isoproterenol to my cells, that behavior increases even more than at baseline. So that suggests that late sodium current facilitated by CAM kinase is a driver for ion concentration homeostasis. And in contrast, the variables that are involved in renin receptor function, such as my SR calcium release or my uptake, have negative impact on those measurements. So late sodium current is a driver for these kind of measurements that I've seen in my mathematical model. So in conclusion, we added a new set of formulations to this well-known human atrial cell model, and we looked to see what kind of dynamics are responsible for the maintenance of atrial fibrillation, and specifically looked at CAM kinase mediation of late sodium current. And we found that CAM kinase mediation of late sodium current is responsible for disrupted ion channel homeostasis and disrupted calcium dynamics in these atrial cells.
Thank you.
Thank you. Is that too fast?